All right, so let's walk through the first lab, investigating factors affecting enzyme activity. You're going to have chunks of potato, and you're going to have hydrogen peroxide as your main thing that you're going to try to work on. The key fact is that the potato, the cells themselves, are going to be our source of the catalase enzyme. The potato cells make catalase, in other words. And the point of the catalase is that it's going to decompose, which is a fancy way of saying break down, hydrogen peroxide. Now, hydrogen peroxide is a molecule that your body makes as a natural waste product byproduct, but you don't want it to stay as hydrogen peroxide within your body, so you'll want to break that one down. And if you were looking at the cell organelle homework already, you would have seen one of those cell organelles was called a peroxide, one of those cells, one of those cell organelles was called a peroxisome, and it's the peroxisomes that break down peroxide so that they can get rid of this harmful peroxide that would otherwise start to interfere with some chemical reactions and cause damage within the cell. So how does it do that? Well, you take hydrogen peroxide, H2O2, we see the per, that means extra oxygen. The catalase enzyme is then going to break it down into water, which is harmless, and oxygen, which is harmless. So when we're looking at the rate of reaction of the catalase enzyme, we should be able to say that if the catalase is very active, then we should see lots of bubbles forming in our liquid, and those bubbles will specifically be made from oxygen gas. If the catalase is not active, then we shouldn't see any bubbles. So there's a couple conditions that we're going to be looking at to say when would the catalase be working and when wouldn't it be. And remember that we're investigating factors based on our lesson, and we said that there are 4 factors that influence it. There's the pH level, there's a temperature level, and then there's the amount of substrate and the amount of enzymes available. And all 4 of those things will determine how quickly these types of chemical reactions can occur. All right? So let's move on now to our very first part. So in part 1, you're asked to take and label 3 test tubes, A, B, and C. And then in test tube A, you need to put 2 mils of hydrogen peroxide. And in test tube C, you're going to put 2 mils of water in there. Now we're going to use C later, and B doesn't get anything just yet. It'll get it in the next step. So once you filled up the peroxide in A, you're going to take a little chunk of potato, a nice fresh piece, and you're going to put it in there. And you're going to see that there's going to be bubbles forming. That means that oxygen gas is being produced, which means we have evidence that the catalase enzyme that's built into the potato is working on the hydrogen peroxide. And in the lab itself, it tells you that how vigorous this reaction is, how many bubbles they're coming, how fast they're coming, is a level 4. So you're told that right off the bat. Then, in step 4, you're supposed to wait for it to stop bubbling. And this is where people usually make mistakes if they're actually doing the practical labs. They don't wait long enough. So if you wait long enough, then you're going to end up having no more bubbles, which means no more oxygen is being produced. And then you need to take the liquid that was in A, and you need to pour it into the empty B test tube. Right. Then you're going to put a new fresh piece of potato in there, and you're going to wait to see if there's a reaction. So you're waiting, and you're waiting, and you're waiting. No bubbles produced. So then you can rate that as a 0. There's no reaction whatsoever. Then in the next step, you're going to take the empty A, which just has that old piece of potato in there, and you're going to add another 2 mils of fresh hydrogen peroxide. And what do you know? You see bubbles again. And so you're looking at it, and you're like, wow, that's very similar to the first time. And so you can rate that as a level 4. The levels, by the way, go from 0, nothing, to 5, which is very vigorous, would be more vigorous than this. Then in the next step, you're asked to take that C that just had the water in it and put a nice fresh piece of potato and wait. You're looking for bubbles. You don't see anything, so you can say that that's also a level 0. And you're going to take these results and you're going to put them into the table, and then you're going to answer the analysis questions for part 1. When you go to part 2 asks you to start heating up a beaker of water. This is just regular water. You don't have to worry about whether it's distilled or not because it's not part of any chemical reaction. You turn on the hot plate and you start to see the steam coming up, and you throw in a thermometer and you see it's about 95 degrees Celsius. Then in step 2, you're asked to make some ice water, and you measure it, and it's about 10 degrees C by the time you've measured it. And then in your third step, you're asked to get some room temperature water, and then you're ready to go for the next section. Then they say, label 3 test tubes, 1, 2, and 3, so as not to confuse them with the ABC you just did. And then we're going to put a nice piece of fresh potato in each of these, and just fill it up with just enough water to cover the potato, because I'm going to take these in my next step and I'm going to throw them into each of these conditions. I'm going to put them into the hot water, into the cold water, into the room temperature water, and let them sit there for 5 minutes. So you just wait for the countdown to end. You want to take them all out at the exact same time, so they've all had literally 5 minutes. Then you can, remembering that number 1 was in the hot, number 2 was in the cold, and number 3 was in room temperature. What you're going to do then is add 2 mils of hydrogen peroxide to each of them. And then when you've done that, you're going to look at how vigorous the reactions were. So in the first one, you're waiting around, and you actually don't see any reaction whatsoever. So you're going to say, okay, well, that's a 0. And you look at the second one, and you're comparing it to the level 4 that you remember how fast it was bubbling before. And you're like, well, that's not as much. Maybe half as many bubbles are kind of coming up at a time. So you would say that's about a level 2. And then you look at the third one, and you're like, well, that looks exactly like the level 4s I had before. So you give it a level 4. And that's the end of part 2. So we've now tested the factors. In this case, we've now tested temperature. Then in our third part, we're going to test the effect of pH. So I'm going to have 3 test tubes. I'm going to label it N for neutral, A for acid, and B for base. So I got a couple mils of peroxide with water, then peroxide with hydrochloric acid, and then peroxide with sodium hydroxide. And before I do anything else, I want to test the pH. So I dip a pH paper into each of them, and I find out that the water's about 7, the acid is about 2, and then the pH of the sodium hydroxide is about, or the base is about 11. So now I have that information. I know which one is acid, base, and neutral. I throw a piece of potato in there, and then I look to see what's the reaction. So that first one in the neutral, again, looks a lot like the level 4 that I saw before. When I look at the reaction in the acid, it's about half. It's less. It's about like the level 2 I saw before, so I'm going to guess that. And then I can see as I'm looking at all 3 at the same time that B, or the base one, is somewhere in between the other 2, so I can call that a level 3. And that's the end of the enzyme lab. You've now done all 3 parts, and you've got your results for all 3 parts. When we move on to the mega lab, which has the 5 mini labs, this is more of a just showing you what the results should be so you can interpret them on your own. So I'm not going to go through necessarily every step that we've done, although there are a few things where I'll show you the steps. So in the first one, basically what they did is they would have taken these Petri dishes. In the first Petri dish, in A, there is a raisin thrown into distilled water. B has a raisin stuck in corn syrup. C has a grape in distilled water, and then D has a grape in corn syrup. All right? And then you would have weighed them before you put them in, so you knew what their mass was. After 15 minutes or so, you would weigh them again, and then whatever the prescribed time is in the lab, you would weigh them a third time because you're looking for a change in mass. And when you go to the overall change in mass at the very end, you would see that the raisin actually has its mass increased ever so slightly in A. In B, the raisin has actually decreased in mass. It's shrunk a little bit. The grape in C, the mass has increased. And then the grape in the corn syrup, the mass has decreased. So you need to be taking a look at those results, and with your lab partners, you guys will discuss why it is that each of the things would have happened here. And you want to think look at the lab title. This is in terms of osmosis. So we're talking about the movement of water, not the movement of sugar. Imagine that the sugar stays on whatever side of the membrane it started on. So if you're talking about osmosis, then you need to be using terms like hypertonic, hypotonic, isotonic to describe which direction the water is going to move. When you look at lab number 2, this is the semi-permeable membrane lab, what you've done is taken a piece of dialysis tubing. It looks a little bit like a freezy tube, but it's not sealed. There's little tiny little holes in this plastic. And basically what you've done is in the first beaker there, you've put iodine in the bag and it looks yellow. In the beaker itself, the water that's in the beaker, you've put some starch solution in there. So it looks white, starch being sugar, a complex carbohydrate. When you look at B, you've done the opposite. You put a little starch bag in there, so it looks white, and then you've put in an iodine in the beaker itself. Then you've done an iodine bag, but just water, and a starch bag, but just water. And what you have to do at the beginning of the lab is come to some kind of a hypothesis to say which of those 3 particles, the iodine, the starch, and the water, do you think is able to move across the membrane? So that's your focus. Which ones can move? And then when you see the results, you're going to interpret that to say, yes, water could or couldn't, iodine could or couldn't, starch could or couldn't pass. So then you can describe the membrane and say whether it's permeable or impermeable to that particular particle. So that's what you want to decide by the end based on the evidence. So what do you see when you finish up this lab? Well, something that looks pretty much like this. These are real results from some decent pictures some kids gave me a couple years ago. So in the first instance, we had the iodine bag. It's now turned clear. And all of a sudden, the starch in the beaker, it was white, fallen at the bottom there, came out of the solution, and all of a sudden it's turned purple. In B, you have the starch bag itself has turned purple. It was white, and the iodine beaker has stayed yellow. Maybe it might be ever so slightly less yellow, but it's still yellow. Then you have the iodine bag has now turned clear. It was yellow, and the water has taken on a slight yellow tinge. And then in the last one, the starch bag started off as white, and it's still white. And the water beaker started off as clear, and it's still clear as well. So what we're looking for there is to see, did iodine move and did starch move? The color changes are going to help us know that because every time iodine and starch are in the same place at the same time, you get this purple color. Iodine is an indicator for the presence of starch. But then the last 2, we're not involving iodine and starch in the same location. We're not trying to look for that. Instead, in C and D, what we're trying to do is look specifically at the movement of water. Now, we can actually see movement of water throughout all of these if we take a look at the mass change. So, in the first one, when you weighed the little baggie, you would have found that it decreased a little bit. Not much, maybe a slight decrease, but you would have seen some decrease in the mass. Okay? In the second one, you would have actually found that the bag weighed more at the end. Then in the third one is a very slight decrease. You may not even have noticed much of a decrease, but there may have been a slight one. And then in the last one, you would see that the mass has increased again. And so now you're going to look at the masses and try to decide which direction did the water go. Now, when you're talking about iodine and starch moving, you need to use the term diffusion, and you talk about how they can or cannot move with their diffusion gradients from high to low because it's all passive, and you decide whether they're able to move or not move and what the evidence is and whether the bag is permeable or impermeable to either iodine and starch. And you're going to use the results in yellow to kind of answer those questions. But when you want to talk about the movement of water specifically, then you want to talk about the mass changes because water is going to make the biggest difference in the mass. Iodine might move a little bit, and if it does, it's not going to contribute much because those iodine particles are very, very small. A water particle is going to be drawn much quicker and have a much bigger effect on the change in mass. Okay? So you may see a slight change in mass if iodine moves or if starch moves, but you'll see a massive change in mass if the water's moving. Okay? So that's your overall results then for that lab. In the next mini lab, we're looking at factors that affect the rate of osmosis. And so you fill up these beakers just full of distilled water, and in each of those beakers you have a little baggie, but inside the bag is a differing concentration of sugar, specifically sucrose, like table sugar. So in the first baggie, you have 0% sucrose, which I hope you know would mean that there's just no sugar. So it's a water bag inside a water beaker, right? Then you've got a 5% solution, 10%, 20%, 40%, and again, that's inside the bag, not inside the beaker. The beaker is just water. So inside the bag, you have increasing concentrations of sugar. And what we're looking at is to see, does the concentration of the sugar speed up the rate of the osmosis? Does it slow it down, or does it do nothing? Those are your three options. Will the concentration of the solute affect how fast or the rate of osmosis? So when you look at your results then, you see that in the first one, where it's a water bag in water, there's no change in mass. Well, that makes sense. However much water moves in is the same water that's going to move out. So overall, you don't have a net change. But when you look at from the 5% all the way up to the 40%, the 5% has a slight increase in mass, and then 10% would be a little bit more, 20% would be a little bit more, and then 40% you have a huge increase in the mass. It's greatly increased. And you have this sliding scale on your way up. Now, if you were actually doing this lab in the real lab and you could get your actual mass results, you'd be able to graph this and do all sorts of stuff. But because we're just doing it in general terms, we're going to skip the graphing. We're going to skip all the percent mass change because we're not able to calculate that without actual gram measurements. And instead, we're just going to talk about, in your lab groups, you're going to discuss the just the analysis questions that you can do based on the theory of osmosis and what you think this is doing to the rate of osmosis. Keep in mind that these bags have all been placed into the beakers at exactly the same time and then removed from the beakers at exactly the same time. So there's no bag here that's spent longer. They've all had the exact same amount of time interacting with the water beakers. Lab number 4, very important for your test. What you would have done is you would have taken these little yeast cells, which are a type of single-celled fungus, and you would have, in the first case here, you would have just left them in your little test tube. In the case on the right, you would have put them into hot water and boiled them for a while. And so then what you would do is you would take a bit of this dye called Congo red, and it's actually a toxic dye. You wouldn't want Congo red to get into your body. You would have to wash your hands and stuff because it's a carcinogen. Well, these little living yeast cells, they don't want it inside them either. So if they are these yeast cells here, what they're going to do is the dye will be able to diffuse into the cell because it can pass into the cell, but the cell is able to actively pump the dye back out again. So when you're looking at these here, you're seeing the results would be that on the left-hand side, you have non-boiled yeast cells, and then you would see that there's no Congo red dye inside them. And then the boiled yeast cells do have Congo red inside. And then you have to come up with why is it that the non-boiled can pump it out, but the boiled ones cannot. And the hint is in the title of the lab, Active Transport in Living Cells. So I'll let you guys run with that one. And then the very last lab that you would have done would have been looking at plant cells. In the case on the left, you just have an onion and normal fresh water, and you're looking at it and you can see the cell wall structures, those little darker spots you can see are the little nucleus in there, but you can't really see a whole lot else going on. I should say that the very dark things that you see on there, the almost black things, are just water bubbles, so you can ignore all of that. But you're just looking at it and it just kind of looks like a normal cell. But when you look over at the onion cell that has been in salt water, you would have dripped some salt water on the slide and looked to see what have happened. You can actually see you've still got the cell wall, but now there's this gap. And now you're seeing that you've got this whole section here has pulled away from the cell wall. And you can see that in every cell there's a gap here. So how do we interpret that? Well, first we would say for our results that the cells on the left look normal, but the cells on the right that cell membrane and the vacuole appear to have pulled away from the cell wall. And then it's up to you to explain those results. All right? So that's the end of the brief overview of what you should have done and seen in those labs. Now it's up to you and your lab partners to meet in the groups I'm about to put you in so that you can discuss these results. I will show you just now